Thanks, Jessica, and thanks everyone for coming out today. So studying context is a really great way to learn about meaning and function. So if you come across a word that you're not familiar with, it's really hard to figure out what it means without first seeing how that word is used in a sentence. But as soon as you start to see it used in even just one sentence, you can start to develop some theories as to what the meaning of that word is. Some of you might have guessed by now, just based on how this word has been used, that slongt has something to do with eating and drinking, and indeed, it means to eat or drink something really fast. So today, I'm going to be talking about how we can use context to better understand the lives of a very enigmatic group of organisms known as fungi. So one challenge for studying fungi is that they're pretty inconspicuous in the environment and really easy to overlook. And because they're so easy to overlook, our interactions with them can sometimes feel pretty random. But with the right tools and techniques, you quickly realize that far from being randomly distributed, fungi actually have preferences for where they do or do not grow. And understanding the genetic bases of these preferences is going to be a critical issue in the upcoming century because we often find fungi growing in places that we'd rather they didn't, such as our food and our forests. Fungi represent some of the most devastating pathogens of both the agricultural crops we eat and the forests that we enjoy. Learning more about the genes that let fungi grow on particular hosts is going to be really instrumental in designing management strategies to kind of take advantage of these preferences and manipulate these preferences, and also to help breed plants that can better resist fungal infection. So one important way that plants resist fungi is through the production of small toxic molecules. These toxic molecules often poison the fungi or just stop the infection dead in its tracks. But some fungal pathogens have evolved these counter-resistance pathways that take these toxic molecules, detoxify them, and break them down. And by doing this, this allows these particular fungal pathogens to kind of break through plant defenses and cause disease. Unfortunately, we don't really know the identity of a lot of the genes in these counter-resistance pathways, and finding these genes is actually quite difficult using currently available software. And this is a problem because if we don't know what genes underlie these important counter-resistance pathways, we're not able to study how these pathways evolve, and we're not able to develop ways to predict and prevent their evolution. So one solution to this is to develop new ways to identify these counter-resistance genes. And this is where context comes into play. So just like words in sentences, genes in genomes are linearly organized along strands of DNA molecules. And just like words in sentences, sometimes if you know the context of a particular gene, it can tell you a lot about the function or predicted function of that gene. One really great example of this is a metabolic gene cluster. So this is a group of neighboring genes that all encode enzymes participating in the same metabolic pathway or process. And in fungi, clustering is actually pretty rare, but it seems to happen most frequently with genes involved in important ecological interactions, potentially like those involved in counter-resistance metabolism. The really great thing about clusters is that if you have compelling evidence that a group of genes is found in a cluster, then you can infer that somehow the functions of those genes are related to each other. And so we use this logic to learn more about unknown genes that could be participating in counter-resistance metabolism by identifying clusters that are associated with genes known to participate in counter-resistance metabolism. So what I did was I developed an algorithm that identifies gene clusters by testing if a particular gene has maintained its association with its neighboring gene for unexpectedly long periods of evolutionary time. And if this group of genes has maintained their neighboring associations and they've maintained their context for a really long period of time, then that's pretty good evidence that these genes are somehow being co-selected for. So what we did was we took this new algorithm and we applied it to a data set of 13 genes known to participate in counter-resistance metabolism. And we tested whether or not these 13 known genes are actually part of larger conserved gene clusters. And we looked for evidence of clustering in a pretty big database of around 550 fungal genomes. And what we found was that these 13 genes are actually found clustered with hundreds of other genes that have a lot of the canonical functions you'd expect for genes in counter-resistance pathways to have. We then looked to see how these genes are kind of organized in fungal genomes, and we found that these thousands of genes are actually organized into 59 distinct types of gene clusters, many of which have never been identified before. And so over here, I've organized the genes into a network. Is there a way to kill the lights? Maybe that will be how people see. So I've organized all of the genes that we've found to be clustered into a network here, where each node in the network represents a particular gene, and each connection between nodes represents an instance where those genes are found clustered in fungal genomes. Once we identified our set of candidate counter-resistance genes and identified some of the clusters that these genes are found in, we looked for patterns in how those clusters are distributed in different fungi. And we found that one subset of clusters are significantly associated with fungi that have plant pathogenic lifestyles, while another subset of the clusters were more associated with fungi that degrade plant material. And this is really significant because it's kind of hinting that these clusters are encoding pathways that let these different types of fungi grow on specific types of plant materials. Another interesting thing that we found was that among all these 59 different distinct types of clusters, there were a group of around 50 genes that were shared between very different types of clusters. And this is exciting because it kind of gives us a clue that maybe these genes have been repeatedly recruited to different types of counter-resistance metabolism. So studying the evolution of these genes in particular is going to be really important to kind of picking apart how these pathways are evolving. So after we took a look at the big picture of counter-resistance metabolism and clustering in counter-resistance metabolism, we decided to take a closer look at the dynamics of clustering as they're happening at the individual genome level. And so we focused our analysis on a particular set of clusters known to associate with a counter-resistance gene known as stilbene cleavage oxidase, or SCO. When we took a closer look at the types of clusters that SCO is found in, we found, yeah, that's great, thank you. We found that SCO is associated with 3 distinct types of clusters, and I've highlighted those clusters here in 3 different colors, in blue, yellow, and green. We started studying how SCO's association with these different types of clusters has changed over time, and instead of finding that SCO has maintained a static relationship with each of these different types of clusters, we actually found a lot of evidence suggesting that SCO is actually switching context very frequently and associating with very distinct types of clusters at very distinct evolutionary time points, sometimes associated with host switches. And here's one example of that over here. So on the left over here, we have a phylogenetic tree showing the relationships between different types of fungi. And on the right over here, we have a little schematic showing how SCO is organized in those fungal genomes. So highlighted in red is SCO, and highlighted in the other colors, so all the accessory genes up here are highlighted in blue, and down here they're highlighted in green, going back to those different types of gene clusters. So in the majority of fungi in the top of the tree here, they're fungi that you'll typically find decomposing plant material out in nature. And in their genomes, you find SCO clustered with this blue gene. But suddenly, down here, in this lineage of grapevine pathogens, you see that SCO suddenly switches context and is now associated with this group of green genes. And when you have a look at the functions encoded by either SCO and the green genes or SCO and the blue genes, you can tell that they're encoding very different types of metabolic pathways. And so the question here that I'm going to be really excited to try and figure out is to see whether or not this pathway over here encoded by this green cluster in SCO is somehow helping this grapevine pathogen grow on its grape host. So just to wrap up, studying the context of genes in genomes is a really great way to learn more about counter-resistance metabolism. Once you've identified that those genes are in metabolic gene clusters, you can then look for patterns in how those clusters are distributed across different types of fungi and start to develop some hypotheses for how these clusters could be contributing to host specificity. The next step for this data set is going to be to try and functionally characterize some of these genes that we've identified to see and to test if and how they're associated with counter-resistance metabolism. But in the meantime, I'm going to keep having a lot of fun taking kind of really in-depth looks at how these clusters have assembled over time so we can learn more about how these different counter-resistance pathways are helping fungi grow on specific host plants. And with that, I'd like to thank everyone involved in this research, especially my mentor, Dr. Jason Slott, as well as all the people who've made their data publicly available and our funding sources. Thank you very much.